How are we doing, Barry?
Go ahead, anytime.
Oh, okay, great. Well, thank you very much for joining us tonight for the BC Bee Breeders' second talk of this year's education series. Just to let you know as well, the talks are recorded. We have a YouTube channel, thanks to Barry, and all the talks are available there. And if you could join that channel or follow it, that helps us support what we're doing, and we can see if you're enjoying it, and it will give us an encouragement as well, so that go ahead and follow the channel if you do go to it. We're also enthusiastic by how much interest has been shown both by BC participants and across Canada, so that's great. If you have any input, you can always send us an email to our secretary and let him know if there's any other talks that you'd like to know or areas that you'd like covered by our education series, especially the members. We're always out there looking for who we should be inviting. But and you'll see here at the bottom of the web page that you can go to for the videos. And with that, let's get on with our second talk. Hygienic Behaviors and Varroa Resistance, a study of selective breeding colonies from Quebec. It's a talk given by Mary Lou Morin, and we're very pleased to have her here to help. I think for me, this talk will help clarify all the different traits that we know about and test for, and which ones might be the most valuable in looking for Varroa resistance. Thank you, and let's let Mary Lou start.
Hello, everyone. Thank you so much, Liz, for inviting me to this series. I'm very happy to present to you the result of my master's project in biology that I did at Université Laval, and to explain a little bit more of these hygienic behaviors that we hear talk about so much. And I just want to preface this by saying that it's 10 p.m. here, so I'm a little bit tired. If I slur my words a little, don't mind me, but I will try to give you the best presentation that I can. All right, so before we attack our subject, I'm going to present myself a little bit more, because I'm sure most of you have never heard of me. I've never seen me present since I'm from Quebec, and I'm pretty early in my career. So since last year, I'm a research professional in agricultural sciences at Laval University in Quebec City. I've completed a master's degree in biology in 2022 and a bachelor's in biology also. And my first introduction to beekeeping and the beekeeping sciences was back when I was an undergrad. I had an apprenticeship with CRESAD, so the research center in Deschambault. I fell in love with it, and I've been working with bees since. So for today, the plan is to first just touch on the subject of hygienic behaviors. We're gonna see what they are, how we measure them, gonna give some examples, and I'm gonna show you also pictures of how we proceed here to test these. Then in another section, I will talk more in depth about my project. And lastly, I'm going to talk about a little bit how we see the future for hygienic behavior in our research center and in the prospect of selective breeding. So let's go ahead. I'm going to ask a very fundamental question here, but how do bees know how to be hygienic? What I'm saying by that is, we see the bees, working all these different tasks from across all different age, all in harmony. So the human way to look at it must be to think maybe the queen is like at the top of the pyramid and explaining to each and every little worker how to do their job, clean this, go seek food. But we know that that's not how it works. In fact, bees are more like little computers. They receive input, process it, and give back output. Like their computers is their genome, and they're just receiving information and giving back behavior. So we're going to take a look at what happens from the stimulus to the externality of the behavior. So our little worker bee here is receiving a lot of stimuli, both from inside. So it can be, depending on its developmental stage, internal physical stimuli, such as hunger. It can be from hormones, and also from its genetics and epigenetics. And there are also external factors that come from the hive or the exterior. So there are sounds, vibration, there's light, there's a magnetic field, and there are also chemical compounds, which are what is most interesting in hygienic behavior. So there's a community of stimuli that are bombarding these workers, and they are perceiving some of them. So they have a multitude of organs to detect, perceive these stimuli. For example, for chemical compounds, we think about the antennae, which have many little sensitive that look like hair, and these are connected to neurons and to the brain. So upon receiving a stimulus, the bee is then processing it in its brain with its molecular transduction. So depending on what genes they have, they're going to emit certain chemicals back, and this is going to translate into the behavior. So let's keep this fundamental mechanism in mind when we think about the hygienic behaviors. Now, hygienic behavior, what is it? It's first a behavioral sequence. So like I said just now, we first have to detect a stimulus. So in the case of hygienic behavior, it's the stimulus that is emitted from the brood that is either dead or parasized in the case of Varroa. So our little worker bee here doing his normal things, we're just walking on the brood surface, detecting under the cap that something is wrong. And then the second step of the sequence is to remove the affected individual. So like I said, it can either be a dead pupa or larvae or parasized. And I like to compare this to the detection and removal of fecal matter from the baby. Why do we have these senses? It's a way to protect ourselves and the people in our nests from disease. And it's the same for the bees, except that they are just putting their babies out of the hive in this case. Hygienic behavior is also a heritable resistance trait. So it's a behavior, but it's a trait that offers resistance towards pathogens and the parasite Varroa. Just a touch on the difference between tolerance and resistance. So I've included this lovely picture from an article from Fanny Maudet. Tolerance is when the host learns to live with the presence of the parasite, but the impairment that is caused by it is reduced. So, for example, in the case of Varroa, there will be as many Varroa present in the hive they will not impact the reproduction of it, and they would just be less affected by its effect, for example. So the traits that are tolerance based are like self-medication. For instance, with the propolis. And when we talk about resistance, it's really to reduce the parasite's fitness, for instance, its reproductive cycle, to keep it below damaging threshold. So to keep it low in infestation level. So this is where the hygienic behavior and also grooming behavior, Varroa sensitive hygiene, and all of these other traits that we're going to touch on later in this presentation. And the fact that it is heritable is highly important. This is what permits us to breed for the resistance, is the fact that it's written in the genes. So it is in the genetic sequence of the bees, and it can be passed to future generations. What differs between an unhygienic and hygienic bees, beside the genes? Mechanistically, the hypotheses that have been tested and are accepted about hygienic is that non-hygienic bees that are not able to effectively detect and remove the brood from the colony, they perceive the same stimulus, but they are not able to either perceive it and or act upon it. So their sensitivity threshold is higher. And if you look at the graph that I drew here, so for instance, we have intensity of the stimuli in the y-axis and time passing in the x-axis. So we have the same intensity in both cases, but in the case of the unhygienic bee, the threshold is too high. So the stimulus does not spark the behavior in these bees. Versus the hygienic bee is highly sensitive, so it's going to react to a much lower stimulus, and this is where you're going to see a quick response from them. So same stimulus, different threshold equals different effect. So this is nice and all, but how do we quantify hygienic behavior? You know, when we do science, we have to have numbers to compare, and we need to have efficient tests that are reliable, and we can repeat them. And oh, someone is drawing on my screen. Yeah, so we can observe it with our own eyes as beekeepers sometimes, but what we need is really an efficient method to quantify it. So I don't know, John D, if you can stop writing on my screen, please. Don't know what's going on with that. Thank you. Okay, I'm gonna go ahead. So, like I said, since the hygienic behavior is the sequence of detecting and removing dead pupa, what we can do is control the killing of a small part of pupa in the colony and look if our bees are able to remove these pupa.
I'm sorry about that.
No problem. Thank you. So what we do is we have to detect the pupa from a certain age. So here we have little pupa with the white coloration and the pink eye. So it's the perfect stage to detect the hygienic behavior. We're going to, for this method, add our little containers, and then we're going to pour liquid nitrogen in as to instantly kill the brood by freezing. So we have a known portion of the brood that is killed. We put this frame back into the hive, the test colony, for 24 hours, and then we take it back and we look how many are still intact or are partially removed by the bees. So for example, here I have a picture of a brood frame after 24 hours of incubation in the colony. And you can see in both circles, there are still quite a few uncapped pupae and still capped cells also. So we would qualify this colony to be not very hygienic. So we can count actually the percentage of hygienicism of this colony versus a very, very hygienic colony. So when we say there are 95 to 100% of the brood that is removed, these are hygienic colonies, because the importance is the time frame at which they are able to remove effectively this dead brood. So the quicker, the better. There's also another test that we have done. I've done this for my master's project. It's the same principle as the other one, but it's called the pin kill brood method. So it's mostly used in Europe in breeding programs, and the main difference is that the pupae are not killed by cold, but they are killed with just a small small perforation with a pin. So we are going to mark our brood to be able to trace back where we made this little holes. And we come back also sometime later and we take the same measurements. So here we count, there are many more pupae that have not been cleaned in this hygienic colony versus this one, where we can see almost all of them were emptied out. So this was the different tests for the typical hygienic behavior. So we're looking exclusively at removal of dead brood. And if we want to look at more specific resistance traits, such as Varroa sensitive hygiene, we have to do more specialized tests. So Varroa sensitive hygiene, also called VSH, is a form of hygienic behavior that specifically targets and eliminates brood infested with Varroa mites. So it's the same sequence as before. The bees detect the presence of the parasite reproducing in the cell, and they are going to open the cap and remove this pupa. And it is very important that, since the behavior that we're looking at, Varroa sensitive hygiene, is the removal of the mites from the brood, we have to test and measure this removal of the mites. So we can, it's a little bit more tricky than to just freeze a part of the brood. I'm going to show you now a schematic of our method. So how we did this assessment. So the principle is that you have a frame of brood with a lot of Varroa mites inside, because you have to see how much the bees are able to remove from this frame, right? So first, you need highly Varroa infested colonies in which you put an empty brood frame. You cage a queen on this frame so that there is uniform laying of eggs on it. And you leave it in this colony until the brood is capped. So when you take this frame out of the colony, the Varroa are trapped inside. So this is what I show here in the second step. We have developed a frame that can be separated in 2 halves, so that for one Varroa-infested colonies, we can test 2 colonies as to be more efficient in our testing. So you have to do an initial evaluation of the brood frames. What we did is we opened 150 cells of each half frame before we put it in our test colony to have a percentage of infestation, initial infestation of our mites inside the brood frame. Then we inserted it in the brood chamber of our test colony, leave it as is for 7 days, and then we were able to retrieve it. And these frames were frozen until the final evaluation, where we checked again for 150 cells to get another infestation level. So by comparing these 2 infestation levels, we are able to see the change. And if it has diminished, then you can say, okay, the bees were able to remove some of the Varroa and to quantify it. And the same frames were used, I will explain to you later what it is, but they were used to extract the recapping activity measurement and the mite non-reproduction measurement also. Now, mite non-reproduction, also called, before it was called suppressed mite non-reproduction. You may have heard of SMR. Now we use MNR. It's a terminology issue because suppressed, researchers are saying that suppressed needs to be reserved to when the brood is actively suppressing the reproduction of the Varroa inside the cell. So when we observe lack of reproduction or diminished reproduction from the Varroa, we call it MNR now. So that might be a little bit confusing if you've heard of SMR before, but just assume that it's basically the same as you've heard before. So what is it? If you've never heard of it, is the failure of a founding Varroa to produce at least 1 mated adult female. So when a mite penetrates a cell, it will reproduce and produce at least 1 other female that will be part of the population in the colony. So different things can happen to say that Varroa is not reproducing. There can be either no eggs. It can lay only 1 male. So if there are no females, then no progeny can emerge. If it lays only females, then these females will not be mated. And even if they enter the colony, they will not be able to reproduce later. So they don't really count as reproducing Varroa. And if they start to lay eggs later in the host, in relation to host development, then it might not be able to produce 1 viable female in time. And how we are able to observe that is by collecting a brood frame and looking through a stereo microscope like this with in a laboratory. It's very time expensive and very minutious work, but we are able to differentiate it. There is a lovely chart made by Buckler. You can find this on the internet, and it states basically, depending on the pupa stage, what you should find in a cell with a Varroa reproducing normally. So if you have a pupa with the dark purple eyes, then you're supposed to find the Varroa mother, one son like this, and at least one female supposed to look like that. So we use this chart to assess the reproducing status of the mite. Another trait that is very interesting for Varroa resistance is called recapping behavior. So recapping is a behavioral sequence that consists of targeting, opening, and then recapping the root cells, leading to the potential disruption of the mite reproduction. So in the VSH, we have the removal of the pupae, and recapping, it opens the cell, but it decides to, for a reason that we are not sure of, set it back, recap it. And the way that we are able to assess this is by opening the cell cap, turning it upside down. And what we can see in this picture in A is the shiny silk cocoon made by the larva for its metamorphosis. And when there is recapping behavior, what you can see is a little hole or a bigger hole left in the silk cocoon by a worker. So there was a disruption of the operculate. Now I'm going to speak a little bit about the research project I did for my master's. The title is Mite Non-Reproduction, Recapping Behavior and Hygienic Behavior are linked to Varroa destructor infestation levels in selected Apis mellifera colonies. And I'm proud to announce that my research is going to be published in Journal of Veterinary Diagnostic Investigation. So it's in process. But you can also find, if you're looking on the Laval University website, you could find my memoir under the French name listed here, but there is a draft of this article that's going to be published. And I did this research with Pierre Giovinazzo, who was my professor, director. So back in 2018, when I started, of course, there was already the threat of Varroa. We were seeing increasingly high overwintering losses, as you are all very aware of. And we were looking left and right for solutions for this. And one of the better fitting solutions for us was to really look at resistant traits and include them potentially in our breeding program. So we were able to do an exploration of VSH and the other resistant trait. So this is what I'm going to present to you today. The objective of my project was therefore to improve the knowledge that we have of the links between these behavioral traits that I've talked about earlier, and their relation to Varroa resistance and social immunity within the ULaval and Cresat bee breeding program. So all these traits, how they relate to Varroa resistance, and how we evaluate Varroa resistance is by looking at the infestation level of Varroa in colonies. So I measured two sets of variables. I like to separate them for comprehension. We have the variables that are measured in a normal setup, I like to say, and then there are the performances in the high mite infestation level setup. So for the normal setup, so it's just not manipulating the hive in any way, is we have hygienic level twice. So first with the free skill brood assay, and then with the pin kill brood assay, like I presented you earlier. And then we measure the natural mite infestation level at the end of the season. So for us, it's mid-August typically. Then for the experimental high mite infestation level, we have the Varroa sensitive hygiene with the change in brood infestation rate, mite non-reproduction, and the recapping behavior. For the methods, I've described 2 methods for basically all of these tests, except for the infestation level of Varroa, we measure them by ethanol wash. So we take a sample of bees in ethanol, and with various shakes, we are able to count the number of mites and relate it as a percentage of infestation of mites per bees, per number of bees. We've repeated the test twice during the season with 2 weeks between each, because we know that these tests give different results depending on different environmental conditions. So it's important to repeat it at least twice during the season and do the average of those 2 results to give a more reliable picture of your colony. And for the experimental setup with a highly infested brood frame, we were not able to do repetitions, but if you want to do this at home, or not at home, but because it's too resource intensive and time consuming, but if you're interested in studying this, I would suggest repeating the test also at least twice to get a better look at the expression of these traits. The first results I'm going to show you is just a summary of the measurements we did. So we have all the variables that we measured, the mean expressed into all of the 34 colonies that we tested, and the minimum, maximum, and the standard error around the mean. So for hygienic testing, we have the 2 tests, free skill and pin kill. The average of the free skill is higher. This can be explained by the time difference between the 2 tests. So free skill, we tested it after 24 hours, and pin kill only 9 hours later. This was because if we waited 24 hours for the pin kill, we would have seen too little variation between the colonies, so too many of them will have been 100%, because we did the tests prior to the real thing. So we found that 9 hours was the best to have a nice range. As you can see, we have a good range of then for VSH, we have I would say pretty high average of Varroa sensitive hygiene. So this is the percentage of reduction of infestation in the brood. Minimum of 0, so no reduction, and maximum of 88%. Pupae removal, I've not talked much about it, but we have also analyzed photos before and after introducing these highly infested frames in the colonies. Not going to talk much about it further. For mite reproduction, non-reproduction, about 31% average. I would say it's pretty medium. Above 50% is considered high. normally. And then for recapping activity, I've separated the measurements into two. So we have the recapping activity of all the cells that I looked at. So if they were infested or not infested, I'm including all the data of this. And under this is the recap cells infested by Varroa only. So the percentage of recapping in the cells that were infested by Varroa. So this is a measure. The difference is to see if the behavior is specific to Varroa or not. And then for Varroa infestation, I would say that we have pretty low infestation level, so averaging 0.6 Varroa per 100 bees. What I did is that I did correlations. So we have a matrix of correlation coefficients here with all our variables listed. So what you can see is the correlation between the two traits at the intersection. And just a reminder, the coefficients go between -1 and 1. So the closer to either 1 and -1, the stronger the relation is. And the closer to 0, it means there is no relation between the two characters. So if it's +1, the two traits are going in the same way. So when one goes up, the other goes up. And when it's negative, it's the opposite. So when one goes up, the other goes down. And what we found is that only 2 of these combination of traits were significant in the correlation. So first, VSH was negatively correlated with recapping. So the more recapping we observed, the less VSH were the colonies. And then the other relationship was more recapping of total cells, the less recapping of only infested cells. So this is really interesting, I found, because what we can see is that it's almost as if too much recapping can be detrimental to the resistance to Varroa. It's as if some bees just uncap and recap, spend a lot of time just uncapping and recapping, but are not able to conclude the behavior. So this is how I would explain or approach this result. And then to go further, I put the resistance rates in relation directly to mite infestation. So I wanted to see if we could predict lower infestation in colonies with either one trait or a combination of the traits. And what I found is that 3 traits were significant to predict Varroa infestation in August. So the first one was mite non-reproduction, when there were, so here in the y-axis is the number of mites per 100 bees, so the infestation level that goes from 0 to about 2.5%. And here is the proportion of mite non-reproduction, so the number of mites that were not reproducing well. And what we see is when MNR is high, the infestation is lower. And inversely, when the proportion of non-reproducing mites is lower, typically we will see more mites in these colonies. The second one that was significant in our model is the recapping of all cells. So like I said, for the correlations, this is going in the same way. So the more recapping and the more Varroa we are expecting to find at the end of the season. And the last one, which I find very interesting also, is the hygienic behavior measured by the free-scale brood method. This is the method that we are using in our breeding program since 2010, and we can see that the most hygienic bees, they are the less infestation we're expecting to see in these colonies. So what we're taking from these results is that mite-node reproduction, recapping, and hygienic behavior by free scale seem to be the most relevant traits in our colonies. But there are still many questions. For example, what's the real significance of recapping behavior? So definitely more research needs to be done to know for sure where to focus our energy. So now I'm going to quickly talk about the perspective that this has brought to our selective breeding program. And before I talk about this, I just want to present to you a little bit my team, who I'm working with, both from Laval and Cressan. So, of course, Pierre Giovinazzo, the professor titular of the CLE, so the chair de leadership enseignement sciences apicoles. Pardon my French. We have Ségolène Maucourt, who's completed her PhD last year on the selection breeding program. And André Rousseau also is also very implicated in this program. So, so you know now that when I say we, I'm talking about all of these people, not just myself, of course. Just to situate you, we are in Quebec, more specifically in Deschambault, in the Portneuf region. The Centre de Recherche en Sciences Animales de Deschambault, so the Deschambault Research Center of Animal Sciences. It's a private center. They have an apicultural science division, and it's in fact the only research facility in province of Quebec that does research on honeybees. We are managing between 400 and 500 colonies for research, and we have apiaries between Quebec City and Deschambault. And yeah, we are doing selective breeding since 2010. Also rearing our own queens, and André Rousseau is our artificial insemination technician. We don't do many, but we are able to include some artificially inseminated queens in our program. If you would like more information about what I'm going to talk about in regards to our selective breeding program, I suggest that you go check out these two papers by Ségolène. But just for now, the traits of interest for us are really hardiness, that translates to winter weight loss, so how much bees consume during the overwintering period, and also the spring development, how fast they can grow in spring. We also have the production of honey and two health traits, which are hygienic behaviors and Varroa destruction infestation. Varroa destructor infestation, sorry. So this is the scheme that we are using. We have about 100 colonies that are evaluated for a whole season on these performance traits. They overwinter. We calculate the breeding values using blob animal model. to select the best colonies of these 100. So about 10 of them go for drone production. Between 7 to 14 of them go for queen rearing. We have our mating site at the farm. So we produce queens, we produce nukes. They are kept another winter, and the following one, we are testing these colonies and we're repeating every year. So we have a rotation of 2, I would say, packages of lineages that are tested each another year. And this is just one of the results that Ségolène has been able to compute. Since the beginning of the selective breeding program, we've been able to improve hygienic behavior by 0.3% per year since the beginning. Moving forward, what we want to do is maintain this breeding program. This is inestimable a richness, I want to say, like it's our jewel. And we want to act as the core of the dissemination of this genetics across the province of Quebec and maybe even Canada. So what we're looking at is to then use our local queen breeders as multiplicator of this genetics. So we could send them genetic baggage or breeders colonies, for example, make them rear multiple queens, and then have our local beekeepers harvest the honey of our work from these more than 10 long years of doing all this work. And we want to keep measuring the traits that I showed you earlier, but we are also looking to include more traits, such as recapping behavior, maybe. And we're working on always improving tests, making new tests that are reliable, reproducible, and efficient. And we also want to touch on incorporating genomics into our breeding program, since we are calculating breeding values with computers, but we are only working on the phenotype. So adding the genotype into this would greatly improve our selection, of course. I want to thank Prof. Giovinazzo, the team Cresad, and my colleagues at Université Laval for their support, the financial aid that we've received, and you for your attention. You can join me at my email address here, and you can also visit our website. So I will be taking questions. Thank you very much for listening.
Thank you very much, Mary Lou. That was awesome. Now Emily Huckster and Wendy Gilson will handle the question and answering period. If you want to, you can raise your hand, and Emily will catch you there. And if you want to, otherwise, you can just type a question into the chat.
Hi, Mary Lou. This is Emily. I have a bunch of questions. So until questions start to show up, I'll just keep asking you questions. So I know that honey production and hygienic behavior are not often, often one goes up and the other goes down over time. And you showed that increase in hygienic behavior over the past, I think it was 10 years. And I'm wondering if your honey production, if you've been able to maintain and improve your honey production over that time period as well.
Yes, I don't want to give spoilers for an eventual presentation by Ségolène, but yes, we have seen improvements, great improvements in honey production. And she's also done the correlation between these two traits. And there doesn't seem to be a significant positive correlation, but it's not negative either. So even if you select for hygienic, it doesn't lower the honey production, or production of honey has increased also.
Okay, nice. Fred has a question. You stated hygienic behavior was improved by 0.3% per year. What value is considered successful?
That's a great question. This is nice to have improvements, but until we see actual resistance to a pathogen, I think, so the principle of hygienic behavior testing is that, yes, you increase the capacity of the bees to remove effectively the dead brood, but you have to periodically submit them to the stressors that you want them to be resistant to. So I don't know for sure if a certain number, like if all of our bees are 100% hygienic in 10 or 20 years, we might not necessarily be able to say that, it's done. We have to actually challenge our bees to the diseases and the parasites to know for sure.
And there's another question here, Mary Lou. How has winter survival been impacted?
Sorry, what?
Just a sec. How has winter survival been impacted? Just a sec.
Has winter survival been impacted by the increase in hygienic behavior?
Thanks, Emily.
I will have to, like I said, I will have to check on Segolen's results. I can't remember if she found an actual link between the two. Sorry, I cannot say for sure, but our overwintering losses have been well below the national average every year. I don't know what that says about the success of our program, but this is all I could say for now. I'm sorry.
No, it's okay.
So is there a possible or are you guys looking at using UBO testing in the future?
UBO?
UBO is the chemical that you can paint onto a section of brood and then come back 2 hours later and check essentially how much brood has been removed.
Thank you. Yes, I remember about that. We have not talked about this, but this is surely something that we would be interested in looking at. So like I showed, we have tried experimentally just testing the different sticky strips to look at recapping behavior. So this could be very quick and easy test that we could implement, as well as the free skill that we usually do. So I'm actually gonna check with my team about that. Thank you for the question. I would like to add also that no, we are testing for many more traits also, but we are not able yet to include them in, the index, the selection index, to select for these traits yet. So in selection, you have to focus your energy on a very minimal set of traits to see actual improvement. So we focus mainly on spring development, honey production, and hygienic behavior first. But we are also taking data slowly on propolis production, recapping, like I said, and we have to accumulate data over many years before we can say first that it is heritable, so that it can be transmitted, and that we see value in including it in our index, selection index.
Yeah, that kind of relates to one of these questions. They wondered if hygienic behavior and gentleness, if there's a connection between those.
Yeah.
We are also measuring for gentleness. In fact, thank you. We haven't checked for the relation between the two, but I know that I've seen some papers that were testing and saying, for example, with the Africanized honeybee, that its more aggressive behavior was linked to the more hygienic behavior, I think. But we have not found similar results in our colonies.
Hi, sorry, I just joined, but like, where did they get these diseases from?
What diseases exactly?
Wait.
That's a loaded question.
Do you see any more good questions there, Emily?
Yep. So what method of mite reduction or mite kill are expressed? Are bees removing infested cells, or are bees biting the mites, etc.? Is one of the questions from Steve.
So in general, in our bees, I guess the question.
Yeah. So how are the mites being removed from these hives?
Okay. Yeah. So in general, the mites can be removed.
Or if you're looking at that, are they?
So we are not measuring VSH every year, sadly, because it would be just too much intensive work for us to do on all 100 colonies every year. And so we are limited to mite.
Are the bees really cool?
To mite traps with sticky boards and to Varroa wash. So what we actually do is just monitor the evolution. Very funny. The evolution of the Varroa infestation level. We don't know how our bees do mechanistically to reduce infestation. It could be from grooming. We have not checked that. It could be from VSH or MNR. We are not entirely sure. So as I showed, there is great variation between our colonies, and the main hypothesis that is pushed by most researchers working on these traits is that bees typically need more than one resistant trait to be resistant. So it's more a combination of all of this that gives resistance to Varroa.
Here's an interesting question from Liz, which is, do you see recapping of drone cells?
We've not checked that, but it's
good question, right?
It's something we sure have to look into. Yeah, I was there for the first talk, which focused on drones, and yeah, we definitely have to look more into it. Because the Varroa are preferentially reproducing in the drone brood, so it would be interesting to see if some of our bees are specialized into removing the mites from these.
Quite just to say, quite often with the high VSH, you'll also see, and I don't know if you've seen it, where the drones, they have kind of purple eyes poking out of the cells. They aren't completely capped. Have you seen any of that?
I can't say I've seen that a lot enough to remember. So
Yeah.
I would say no, I'm not sure.
And did you start off with any VSH stock, or have you just been working up with the hygienic behavior?
So I know that we've imported, I think, a couple of queens from Baton Rouge in 2015 or 2016. So there might be some remnants of this genetic, but since we've not been testing extensively and selecting these bees, there's a high possibility that we've lost the initial genetic baggage, I would say. So there was some importation, but we're not sure what's left of it.
Another question that's out there is, in your research, have you seen any reduction to the viral loads in hives with an increase in hygienic behavior?
I cannot say that we have measured that we've seen difference in viral loads, since we have not measured for our breeding program. We have not measured the viral load. We've just started developing the methods to do this, and last year was the first year that we sampled for viral infestation. And to start, we're more interested in seeing if there is a variation during the season in each colony. So we sampled every 2 weeks for viruses and nosema also. And we're going to see if there is a trend during the season. We're also going to test for that this year. So eventually, if you keep this up, we'll be able to answer your question.
I like it. It takes a lot of work and effort.
Yeah, it does. It does.
So many measurements to make. So another question is, in your experiments, how does one argue that hygienic and Varroa sensitive are directly correlated? Does hygienic not also spread diseases in higher infected colonies? So I guess it's kind of two questions.
So to answer the first one, there may be correlated, but they might not be, since the stimulus is very different to initiate the behavior. So when you test for general hygienic behavior, it's probably a much stronger chemical cue, or a cue that the bees are more equipped to detect, since the death pheromone has been present for many more years than the Varroa in our species. So Varroa was it's a new parasite. So there might not be correlated, in fact. So a high hygienic bee might not be resistant to Varroa, if that makes sense. I don't know if I've answered the question correctly. What was the second one?
Yeah, no, I think that answers the first one. And then the second question is, does hygienic not also spread diseases in higher infected colonies? So I guess does hygienic behavior?
Yeah, so the principle of hygienic behavior to be efficient, it's that you have to remove the diseased brood before it becomes infectious. So for example, for chalk brood, there were studies that were made that proved that hygienic behavior was efficient at increasing resistance to chalk brood. It has to be made before the sporulation phase, so before the spore are able to disseminate to the other brood. And we can also imagine that for Varroa, portion of the Varroa mites reproducing that are removed are able to reintroduce other brood cells, but it definitely has an effect on the population overall because it's going to stop the cycle of reproduction, and this mite that has been disturbed might not be able to effectively reproduce on the next one, so it's going to delay. And really, timing is what's most important when we look at the progression of Varroa infestation during the summer, because every 2 to 3 weeks, the population doubles. So if we're able to just slow a little bit the progression, then it's going to have an effect on the long run. But if the population of Varroa is really super high already, maybe it's just that the bees are not equipped to remove effectively. So eventually there might be needing for acaricides in this before it's too late. So it's not necessarily I would say totally resistant like feral colonies.
Yeah, I wondered if some of the results that you presented on the recapping and not having that much effect, I wondered if some of that is because the infestation levels in the colonies you're working with are so low. Do you think that has any effect of the fact that your infestation levels are so low in these colonies that you're using, which is wonderful, but
yeah, it's wonderful in a sense, but yes, as you said, we did not have much variation in the Varroa infestation level, and this is a bit lacking in our study, in fact. But I have to say that these colonies are treated each year. We cannot afford to lose them by not treating for Varroa. So there might be a combination of the behavior expressed and also the efficient management that not to say that if you have a lot of Varroa, you're not doing efficient management. That's not what I'm saying at all, but these are not untreated colonies, so I cannot explain all just by the quality of the hygienic behavior, sadly.
right. Yeah. Somebody asked, are there any thoughts on those bees called ankle biter bees? And I think they're specifically talking about the bees that bite the Varroa's legs off, but correct me if I'm wrong, Gia.
The question was if I had any thoughts about that.
Yeah. Yeah, on the bees that tend to show that trait of biting the Varroa's legs off as a means of Varroa removal.
In truth, I don't have much opinion on this because I really did not focus on this behavior. So I'm aware that I think it's also called the grooming behavior, probably what it's referring to. That it's real. I know that it's real, it's observable. I know that, for example, people in Ontario have been working on this, but I cannot say much more than that.
Okay, and then a question that kind of relates back to the previous comment that you were making about your treatments is what are you treating with and when are you treating? And how many times a year?
So retreating, we are alternating between Simovar and what is it called?
Apivar, Apistan?
Yeah, that's it. Apivar, thank you.
No problem.
Amatras then, yeah.
in September usually, or end of August. And then we complete with the oxalic acid drip, I think. This is not my department, so I'm not 100% sure, but I think that I'm sure that it's oxalic acid, but I'm not sure the method. And then we usually don't treat in spring. So we do screening, but we try not to treat these colonies in the breeding program in spring. Actually, not much of our colonies at all, except if it's really concerning. We have projects on summer treatments, but it's not something that we have implemented in our colonies yet for now. So 2 treatments in fall.
And I have a question, like, do you have any comments to make about the hygienic behavior and AFB, American Foulbrood?
I don't actually. I've seen research made in the early 2000s by Marles Spivak about this. Is it? No, it's European. I'm really not sure. I'm not going to say anymore.
So somebody wants to know if you've seen, and I think this is an interesting question, is there any research to see Varroa preference for old comb versus new comb? Have you noticed that when you were looking at the reproduction of the mites within the colonies? I may be just anecdotal, I guess, at this point.
I have not. No, I have not seen that. It's not impossible, but
Just not seen yet.
no, exactly.
How are we doing with the questions, Emily?
Any comments on why the increase in VSH behavior is as low as 0.3%? So again, I think you were saying that there isn't a, you know,
Or hygienic behavior?
Yeah, a Varroa sensitive hygiene, sorry. So, sorry, any comments on why the increase in Varroa sensitive hygiene behavior is so low at 0.3%?
I'm not sure. I think it's referring to this graph. So hygienic behavior as an average. So these are the breeding values. It's not a copy paste of the real percentage that we see on average, but we see an increase of 0.3% each year since 2010. So that's 3 point something percent overall. It might not seem huge, but I mean, I'm not sure what it's supposed to be. I know that the heritability has been calculated by Segalen, and for hygienic behavior, it's not that high. So that explains why we are not seeing quicker progress. So the more the trait is heritable, the bigger portion of it is going to be transmitted to the descendants. And we know that breeding for bees and selective breeding for bees is incredibly complicated. I wanted to add a slide to speak on that, but you have to account for the fact that when you're free mating the bees, you don't know for sure if it's your drones that are mating with your queen. We are trying hard for it, but we're not always 100% sure. There is high variability in the genetic composition in a single colony. You have all the environmental factors to take into account. So I would say it's pretty good. No, we've seen improvement.
Improvement's always a win, I think.
Yeah,
exactly.
So, and there's one more question here, and I think this is the last question. In your research, have you seen anything that Varroa that suggests Varroa mites are getting stronger in their persistence in the hive in dealing with a hygienic and Varroa sensitive behavior?
So this would be if we're seeing the Varroa responding to the bee response to the Varroa. So if there is coevolution happening in real time,
Yeah.
From Varroas to the bees. We have not seen that because we're not even sure if our bees are, well, we know that they are not adapted to Varroa yet. So until we see that the behaviors are really causing great damage to the Varroa's fitness, and then it has to operationally adapt to it. You know, it's the game of evolution. We've not seen that yet. It's not impossible. We see that happening with the acaricides, but it's a little bit different. It's the host-parasite relationship. So no, we fortunately have not seen a response from Varroa.
And I think too, if you're treating it, it probably doesn't show up as much as well. Maybe.
Exactly.
I could be wrong on that, but yeah. And then there was just a comment that it is your breeding yard in an isolated location.
Yes. Sorry. We were able to actually identify our mating site where the drones congregate and meet the virgin queens. So that's pretty cool. We could eventually go and sample the drones to verify if they are from our mating station. It's fairly isolated, so we've tried to, it's not in a mountain or on an island or anything, but when we set up our mating system, we made sure that there were no beekeepers around, but that's a problem that we think that we could face because there are more and more beekeepers in the area. So we're looking at avenues maybe to limit in a 5 kilometers perimeter around our mating station, maybe to have a special derogation or something. But yeah, we're doing everything we can with flooding, with our drones too, with drone frames, but that's about all we were able to do. And also using the artificial insemination eliminates that problem, but we are not able for now to produce as many queens, inseminated queens as we would need for this whole program for now.
Wonderful. Yeah, I think that seems like you've really looked into it. There is a lot of people, I think, listening this evening who study quite a few feral colonies or look at feral colonies. And there was one more question to ask if you guys have looked at the feral colonies as well in the area. It's wonderful to map the drone congregation areas. And I think they were also wondering if you have looked at the feral colonies in the area as well. They probably only improve the genetics, really.
Yeah, but we have not, sadly, no, we have not looked into that at all.
Fair enough. It's hard to do it all.
Yeah, we're trying, but we need more people.
Yeah, don't we all? I love it. Well, thank you very, very much. I'll let Elizabeth speak over.
Right. Thank you all for your attendance, and Mary Lou in particular for sharing all your research with us and answering so many questions. Really, really happy to have you here giving us a talk on this topic. And I'd like to also thank those that have been behind the scenes and helping with our education series. Dan Mawson, who is our webpage for the BCHPA and the BCB breeders, Barry Denlock, and Wendy Gilson and Emily Huckster, all part of the presentation committee that have made this series such a success. As well, if you'd like to show your appreciation for what we're putting forward, you're more than welcome to donate, and the link to that is at the base of this slide. And our next speaker in this series is going to be Dr. Kira Wagner from the University of North Carolina in Greensboro. And she's going to be talking about the UBO test, as well as, I think, a bit about viruses as well and what they're working on there, some of her newer work. And that'll be March 9th at 7 p.m. With that, I think we've all had a nice long session for this education series. And once again, I'd like to thank you very much, Mary Lou, for sharing your research. Really appreciate it.
Thank you. Thank you for inviting me. It was my pleasure.